So next up we have Codey Mathis. She's going to be talking to us about some factors of native pollinators and regenerating forests. So here you go.
Thank you. Hi guys, thanks for coming today. My name is Codey Mathis and I'm a master's student at the Indiana University of Pennsylvania. And I'm here today to talk to you about some of the research in my master's thesis looking at factors affecting the pollinator diversity and density within regenerating managed forests in Pennsylvania. So it's been pretty well documented that there are widespread pollinator declines across the globe, and particularly in North America. And the majority, there are many factors because of this, and one of them is habitat loss. And the majority of the world's flowering plants, including many agricultural crops, rely on insect pollination, the majority of which is provided by bees. And so they are important for agricultural ecosystem services and the pollination. And a recent synthesis of the state of the North American bee population stressed the need to better understand these populations within non-agricultural settings. And indeed, when we look at most of the pollinator studies within the eastern U.S., we see that they focus on agricultural farmlands and urban landscapes, despite the fact that deciduous forest is the dominant habitat cover type in this area. There is growing evidence that early successional forests may provide optimal habitat for stable populations of pollinators. And this is because younger forests have increased light availability that allow for a more diverse and abundant floral community. Ground is accessible for ground nesting bees, and coarse woody debris is accessible for wood nesting bees. Many of the studies that have been conducted in this area have looked at stands 10 to 40 years post-harvest, but have neglected recently harvested stands. With this in mind, I set out to ask two main questions, the first of which is what habitat characteristics drive variation in pollinator density and diversity within these recently harvested stands, and how does floral diversity, specifically the floral community and the diversity and density of it, drive this variation as well? So in 2018, I surveyed 75 sites across Pennsylvania, 37 of which were public and 38 private. All had experienced overstory removal harvest within the last 9 years, and all of our private lands were enrolled in the NRCS Working Lands for Wildlife program and were created for the golden-winged warbler. All of our sites were deciduous forests and were Appalachian oak or northern hardwood, and these are just three pictures of our sites. The one on the left is more indicative of a younger site, maybe 1 to 2 years post-harvest. Moving to the right, you see one of our older sites. So you can see that it's fairly open in a younger site and fairly closed in the older sites. So to investigate the pollinator community, I used two different survey methods. The first was a visual pollinator survey, and this was a modification of the Xerxes Society's streamlined bee monitoring protocol, in which I walked a 66-meter transect within 30 minutes, and I recorded every pollinator that I observed during that transect. I recorded their behavior, their perpendicular distance from the transect, and any floral associations that they had. All of our bees, since it's almost impossible to identify a bee while it's flying, and there are 430 bee species in Pennsylvania, we identified them to 6 morphospecies, which you can see here, the carpenter bees, bumblebees, honeybees, and then medium native, small black, and small green. And butterflies were identified to species if possible. And then all of our visual pollinator surveys were only conducted in favorable conditions, which is temperatures above 60 degrees Fahrenheit, low wind, and no rain. And we repeated these surveys across the growing season 5 times between May and August of 2018. Our second method to look at the pollinator community was with lethal sampling. So we used elevated blue vein traps and ground level pan traps. And I used these two different methods because they are statistically proven to collect different members of the pollinator community. And all traps were deployed immediately following our visual pollinator survey and collected 24 hours after deployment. And we pinned and identified all of the specimens. To investigate the floral community on our sites, I identified every flower that was flowering at the time of our visual pollinator survey on 1 meter on either side of that transect. And we counted and estimated the number of flowering stems and the number of flowers per flowering stem. And we identified all of our flowers with these field guides and then also the Pennsylvania Wildflowers app and iNaturalist. And then finally, to look at the microhabitat characteristics, the site veg characteristics, we had 3 100-meter transects that radiated from our transect center. And at each 10 meters, we stopped on the transects 10 times every 10 meters to look at the presence or absence of the veg characteristics of interest, and with an ocular loop tube. And so what that would look like is if we stopped at this first one, the observer would look up and down, and they would mark that there was canopy, large saplings, forb, and leaf litter. Go 10 meters further, and you do the same thing. And it allows us to calculate percent cover of these characteristics on our sites. So what did we find? For our visual survey in 2018, we observed over 1,800 total bees and 279 butterflies. The three most common butterflies we saw were the red-spotted purple, eastern tiger swallowtail, and spicebush swallowtail. For floral abundance, we counted or estimated over 316,000 total flowers comprised of 161 floral species. And we collected 773 bees. The three most common genera were Ceratina, Dialectus, and Lasioglossum, which would have been categorized as small black bees in our visual survey. For our visual surveys, we saw the, so y-axis, we have mean observed pollinator abundance, and then on the x-axis, we have over time, our 5 different survey times. And so we saw the most pollinators in late July. And it's important to note here that our private sites, which were the Working Lands for Wildlife sites, we actually observed significantly more bees at that time. For floral abundance, we saw that it peaked in May, which makes sense because that's when peak bloom is. And then additionally, at the time in May, most of the things that are flowering are going to be things like blackberry or blueberry, huckleberry, things that have a lot of flowers per stem. So that accounts for the really high abundance that we saw at that time. And then finally, floral diversity was also highest in May. And here it is really interesting to note that our private sites, the Working Lands for Wildlife sites, consistently had more diverse floral communities than did adjacent public sites. So when we look at specifically what flowers we saw the pollinators actually interacting with during our visual surveys, we see that they really tracked the abundant floral community throughout the season. This table shows the top 3 most visited flowers during our visual surveys. We see that in May they're using what's there, the blueberries and huckleberries that are really abundant. And then switching to June and July, we see that they are using more of rubus, still using some small shrubs like the laurels, and then some forbs. And then in late July and August, they're still using rubus, switching their forbs to some of the asters. And they're also using a small tree, which is devil's walking stick, which has a lot of flowers at the top of it, and it was a very important floral resource at that time. So some people might notice the goldenrods are missing. And that's because goldenrods bloom later in the season. But we did, in 2019, push our surveys back later, so we did account for that in our 2019 data. So to look at the density analysis, what I was really interested in is determining which of our variables promote the density of pollinators on these sites. And to do this, we used hierarchical distance-based density estimation with the R package unmarked. And we modeled each veg characteristic individually for each sampling round to investigate the significance as it changes throughout the season. And then we did the same thing for floral community metrics and for stand age. And the important thing to note about this is that it accounts for imperfect detection, which is very important when you're talking about a 10 millimeter insect that's flying by you. Every model gets a detection probability and accounted for detection probability, and you can see that on this graph. As you get farther away from the observer, it is more difficult to detect a bee. And so it's very important that this is accounted for in our density estimates. So then here, what do we find? This graph shows on the left the grass forb fern, and these are these are characteristics that are more indicative of younger sites. And then on the right are going to be things that are more indicative of older sites. And I just want you to keep that in mind as I show these results. So what exactly did we find? We found that these younger site characteristics like grass and forbs were consistently positive predictors of pollinator density. And then the older site characteristics like saplings and canopy trees were consistently negative predictors. Ferns were also consistently negative predictors, and this was likely due to the fact that ferns grow in very dense clusters and generally inhibit the growth of other flowers on these sites. And rubus and shrubs was variable across the season, so we're interested in why that might be, and we look a little closer. And we find that in May, those small shrubs, like I mentioned, were flowering and were very important for pollinators. So we see that our models predict that as small shrub cover increases, I have a laser pointer. As small shrub cover increases, bee density is also predicted to increase. And conversely, down here we see rubus in May. As rubus cover increases, bee density is predicted to decrease. And that's likely because rubus is not flowering at that time. Makes sense. Then in August, we see the inverse. So this is when those small shrubs are no longer flowering. And as the small shrub cover increases, bee density is predicted to decrease very precipitously. And rubus cover also increases bee density. So this is important to note because a site that might have been great for bees in May is not necessarily as great for bees later in the season. And this really drives home the fact that we need to have diverse floral plant communities on these sites for our pollinators. When we look specifically at the floral community metrics, floral abundance and floral diversity, no big surprise, they're consistently positive predictors of bee and butterfly densities. And this is a lot stronger of a relationship for bees, and it's because they benefit from an abundant floral community and because they're provisioning not only themselves during their flight period, but also their young. This relationship is not as strong for butterflies, but it is still positive. And that is likely because they are only self-provisioning. They're not actually provisioning their young. They usually lay an egg on a food plant and then just kind of leave their young. When we look at stand age for density, we find that as stand age increases, bee density decreases. And our models specifically predicted that a site 1 year post-harvest had predicted 640 bees per hectare, whereas 9 years post-harvest, that same site would only have 111 bees per hectare. And that means that our models predict that our younger sites have 5 times more bees than do our older sites. And this is likely because those older site characteristics, like I showed before, inhibit the floral community. And yeah, that's really it. Okay, so switching over to diversity. I was interested in doing the same thing to determine which variables promote the overall diversity of our pollinator communities. And to do that, I used generalized linear mixed models, and I ran a calculated pollinator diversity metric for each visit against the scaled environmental variables. And then I did backward stepwise selection to select the best model based on the AIC value, and then ran a goodness of fit test for the top candidate model. So here are the results from that. And I just want to emphasize before I go through these that a red box around the text means that it was a negative predictor of diversity. And this little B symbol right here means that it was a positive predictor for diversity. So in late May, the only thing that was significant was large saplings as a negative predictor of pollinator diversity. When we go to mid-June, stand age is a negative predictor, and rubus and floral abundance are positive predictors. Early July, large saplings and canopy were negative, and floral abundance was positive. Late July, large saplings, canopy cover, and small shrubs are negative, and floral diversity and grass cover was positive. And then August, large saplings, stand age, and small shrubs were negative, and floral abundance was positive. So the general trend that I see here is that positive predictors were more characteristics of younger sites or a more abundant floral community, and the negative predictors were all within our older site characteristics. So again, it's really just driving home the fact that those younger sites are very important for not only not only abundant but diverse pollinator communities. So finally, I was also interested in looking at which variables drive the diversity, but also visualizing how each of these variables are important for the different species within the pollinator communities. So for that, I used a redundancy analysis, and I modeled each sampling round independently. And the best models were selected that had the low variable inflation factors and highest adjusted R-squared values. So here we have that same graphic, but I'm actually going to put up the different species the different morphospecies that had significant predictions. So starting with bumblebees, we see the predicted hypothesized thing that those younger site characteristics, grass, forb, and rubus, were positive, and the negative associations were with ferns, shrubs, saplings, and canopy trees. Moving on to small black bees. We see that they are positively associated with grass and rubus. There actually was no significant association with forbs and ferns for the small black bees. and saplings was negative, and we see that the shrub and canopy trees were actually variable across the season. For small green bees, we again see the expected, hypothesized associations. And for butterflies, we see the same, except for with rubus, it was variable. So really just putting all of this up here, it just drives home the fact that there is no one habitat that is going to be best for all pollinators within the area. So having multiple habitats that have diverse plant communities is going to be very important for a diverse pollinator community. So in conclusion, our older timber harvest sites appeared to have fewer pollinators, likely because of veg characteristics that limit floral resource availability. Native pollinators have dynamic habitat associations with these veg characteristics. It's not always positive, not always negative. So but they do consistently like some vegetation like forbs and grass. So this really just drives home that it's important to have a diverse plant community to support diverse pollinators across the entire growing season, but also the fact that it's just another piece in the puzzle that early successional forests can be important. And six years after you create these sites, they might not be as good for bees, or for pollinators. So having more of those sites on the landscape and having a heterogeneous landscape is going to be very important for not only golden-winged warblers and gopher tortoises and New England cottontails, but for pollinators as well. So with that, I'd like to thank the IUP Department of Biology, Pennsylvania DCNR, Pennsylvania Game Commission, the funding source of USDA Conservation Effects Assessment Project, and finally, Pennsylvania landowners for letting me tromp around in their timber harvests all summer. And with that, I'll take any questions.
We do have some time for a few questions.
Yes. Yes.
How far down did you take the species?
Yeah.
So, in your local species associated with different habitats, do you have species associated with those different habitats?
Yeah, that's a. Yeah, so the question was about, I was showing the morphospecies data, but do I have species-specific data for the different species within the morphospecies? Is that basically your question? That is the next stage of my analysis. I do have all of my specimens identified, and now I just have to run that same exact analysis for the individual species.
What you're talking about density, you're actually measuring activities. And so what you're going to get potentially between different habitat types is different flying capabilities of bees.
Mm-hmm.
So the probability of encountering a trap increases with flight capability.
Mm-hmm.
So by saying there's more on private and less on public, could be a faction or a function of what particular species are associated with rather than an actual true density.
Okay.
Something to think about as you go ahead. You have a comment?
Yeah, thank you.
Anybody else? I've got one since we have a minute.
Yeah.
Jeff showed a slide earlier where it takes a year or two before golden-winged warblers show up on these openings that are created. And you showed a slide where pollinators show up initially and then go down. So is it fair to say that these openings, while they're waiting for golden wings, are great for pollinators?
Yeah, that's actually a great point, and it's one that we illustrated earlier in the year. We actually published this information for the Pennsylvania Forest Landowners Association at a conference, and we showed that these sites are wonderful for pollinators up until 6 years, and then around the time that they start being not so great for pollinators, they're actually great for golden wings. So these early successional forests are not only beneficial for one species, but for multiple species, and I think that's
It depends on what time in the phase you are in.
exactly. So it's important to have multiple ages of these sites across the landscape, and I think you had mentioned it during Jeff's talk that the creation of this habitat throughout throughout time is going to be very important for all of these species. Definitely.
Okay. Thanks.
Thank you.